Hey, it's Mr. Peterson. Just a short little screencast about polymerase chain reaction. So, some important things here just to look at. So, PCR, polymerase chain reaction. So, what is polymerase and what's a chain reaction? So, here's a short little video about that. So polymerase chain reaction basically takes a little bit of DNA and makes a whole lot of DNA. That's really the whole point of polymerase chain reaction. And how you're going to do this in class with a couple of different labs is a good demonstration of this tool and technology. So, kind of the steps of amplifying a DNA sample, and these are all really simplified, but we're going to wind up with DNA polymerase. So, what's DNA polymerase do? That's right, it makes more DNA. So it reads the code, produces another strand of DNA. Well, if we have a whole bunch of DNA and a whole bunch of DNA polymerases, and we're going to see that in the experiments we're going to do, we're going to make a whole bunch of copies of DNA. So we start with a little bit of DNA and we just keep making a lot of it. So that's what polymerase chain reaction does, kind of. It's just you pretend this is not really accurate. There are far more details than that, but that's kind of the big idea. All right, so we're going to use something called a thermal cycler, and a thermal cycler um, is a thermal cycler is a device that can change temperature. We can actually program it. You'll see it being programmed on my computer. And what it does is it takes a sample of DNA, heats it up to about 94 degrees Celsius. So that's pretty hot, right? So it's just short of boiling. And when that happens, that denatures the hydrogen bonds that hold the chains of DNA together, the side chains of DNA together, fall apart. Well, then we cool it off and we add some new DNA. So in the little PCR vial is going to be nucleotides, and those nucleotides, along with the DNA polymerase, are going to anneal, which means they're going to stick, and we're going to elongate those strands of DNA until finally we make more DNA. And that is just going to happen over and over and over again. So the ups and downs of the cycle with the presence of spare DNA and lots of DNA polymerase molecules, we can start with one strand of DNA and make bajillions of them. So we're amplifying the DNA. We're replicating the amount of DNA so that we can start with like a small sample you might find like inside a few cheek cells, and we can make enough DNA to analyze with gel electrophoresis. So that's PCR in a nutshell.